Okay, good afternoon, everyone. Can you hear me okay? Great. We'll just give a couple of minutes for the last few folks to come and find their spots. Well, good afternoon, everyone. Welcome. Welcome to close to the first day of spring, something to celebrate, as well as our Walsh lecture today. So I'm Armin Razdahan from the NIMH Human Genetics branch, and I'm delighted to be here to introduce our Walsh lecturer today, Dr. Chris Walsh. Dr. Walsh is the Bullard Professor of Neurology at Harvard Medical School, Chief of the Division of Genetics at Children's Hospital in Boston, and an investigator at the Howard Hughes Medical Institute. And as we'll see together today, the Walsh Lab achieves really an exquisite interweaving of basic and clinical science, shedding fundamental light on how our brains evolve, develop, and age, and through this, better understanding mechanisms of disease in neurology and psychiatry. Dr. Walsh and his lab's work have been recognized by numerous honors and awards from organizations such as the American Neurological Association, the American Epilepsy Association, and the National Academy of Medicine. Dr. Walsh completed his MD and Ph.D. at the University of Chicago with Ray Guillory, and after a neurology residence at Massachusetts General Hospital, he completed a research fellowship in genetics at Harvard Medical School with Connie Kepko. He's held the Bullard Professorship at Harvard since 1999 and joined Children's as Chief of Genetics in 2006. Dr. Walsh's clinical research at Boston's Children's is deeply rooted in team science and collaboration with families. And this work has identified genetic causes for hundreds of conditions in neurology and psychiatry, developmental disorders. And his recent work, as I think we'll hear a bit today, has extended into brain aging as well. And in seeking to understand mechanisms of genetic disease, Dr. Walsh and his lab have tackled some of the deepest basic questions in brain development and aging. What evolutionary and developmental processes account for the dramatic expansion of our folded cortical sheets? What are the lineage relationships that connect cells over the dividing cells over the course of brain development? And how can we use these lineages to tell us about the patterning of brain organization in health and disease? And the tools that the Walsh Lab developed to address these questions really open up new fields of biology, new fields of research, and the rewriting of some textbook paragraphs as well. And the Walsh Lab doesn't only generate a thrilling series of papers in some of the highest impact journals of our field, but also, importantly, large cohorts of wonderfully trained individuals who go on to continue the work in their own labs. So as I'm sure you can tell, we're in for a real treat today, and thank you all for coming. So before I hand over to Dr. Walsh, I just wanted to let you know of a few housekeeping things. So you can submit live questions to the speaker through the Send Live Feedback option on the videocast page, and today's CME code is 50117. 50117. So with that, please join me in welcoming our Walsh speaker for today, Dr. Chris Walsh.
Thanks very much, Armin, for the invitation to come here. It's a real thrill when I look at the list of other people who've given the Walsh lectures. And it's great, of course, to visit Washington. I got a chance to see the cherry blossoms, although from the sort of the back seat of a speeding taxi cab. Anyway, it's great to be here. And so our lab has been interested for a long time in using human genetics to understand the basic biology of how the human brain develops. Just for those of you who might not be neuroscientists, I just want to remind you that the cells in the cerebral cortex, the neurons of the cerebral cortex, are non-dividing cells, and so they are formed during fetal gestation, we think of them as being born on a particular day when they undergo their final cell division and become permanently postmitotic for lifelong. Let me see if I can work out this pointer. So you can see here that the different neurons and the different layers are born in sequence, so that these are the firstborn ones, and the lastborn ones actually end up at the top of the cortex. And then after that, the same progenitor cells give rise to the glia, the astrocytes, and oligodendrocyte precursors. So there are all sorts of the 8 billion people on our planet are really a saturation mutagenesis for every possible gene that's required for that process. But today I'll be talking about mutations that occur not that regulate that development, but that occur actually during that development as well. And so these are mosaic mutations or somatic mutations, and they occur actually with every cell division that generates the embryo. So whenever the genome is copied, the copy is never as good as the original, and there are always a couple of point mutations that are introduced by that, and occasionally things like line element transpositions or copy number variants. And so these somatic mutations occur with every cell division that generates the embryo. They can occur early, like this red mutation, before gastrulation, in which case they'll be present in all tissues, but only in a fraction of the cells in any given tissue. They can occur after gastrulation, in which case they might be limited to the brain, like this yellow mutation, and typically would be in a smaller fraction of cells in the brain than if it had occurred earlier. Or there can actually be examples of mutations that can be in one half of the brain and not the other, as this green mutation, or even in a small region of the brain. And actually, at the end of the talk, I'll tell you about somatic mutations that can be limited to just one single non-dividing neuron. I'm thrilled that some of this work has been supported now by the Common Fund of NIH through the SMOT Consortium, the Somatic Mosaicism Across Human Tissues, or as we call it in Boston, the SMART Consortium. So anyway, these clonal somatic mutations have been an obsession of us for about the last 10 years, and they contribute to a growing number of neurological and psychiatric diseases. And so I'll tell you about some of them over the course of the next 45 minutes or so. I'll tell you very briefly about somatic activating mutations in the Mtor pathway in pediatric epilepsy. Then I'll talk about newer work, most of it unpublished, about temporal lobe epilepsy, and then somatic tumor suppressor mutations in the microglia and the Alzheimer brain. And then at the end, I'll talk a bit about these somatic mutations that are limited to a single neuron. So really, a lot of our work on these somatic mutations started because of the interest of Anne Paduri, a child neurologist who joined the lab now 10 or 15 years ago and wanted to understand what was going on in the brain of this young boy who had one hemisphere, the right hemisphere, although radiologists like to mess this up by showing it on the left side. And that abnormal hemisphere is overgrown and doesn't show very good distinction of the normal gray matter and the white matter, and it's intensely epileptic. And so this child had intractable epilepsy starting at the time of birth, and left-sided weakness because that right hemisphere, even though it was big, it was not doing much good. And so in a desperate attempt to control his epilepsy, neurosurgeons at the Brigham removed his entire right hemisphere. And so what you see here in this white is just cerebrospinal fluid. And everything right down to the thalamus has been removed. And this was life-saving. He had no seizures for 6 years. His seizures remain very well controlled. He learned to walk. He learned to talk. He reads at grade level. And so, he really got his life back from this drastic surgery. And his name, here's his picture. He thinks of himself, Dante is his name. I use his name and picture with his parents' permission. He remains a little weak on the left side, particularly his left arm, and he needs a brace to walk, but he basically is doing amazingly well. And so so we postulated that this might be a mutation that's only present in that one abnormal half of the brain, and sure enough, his mutation is shown here. It's a point mutation in a gene called AKT3. It creates a specific amino acid substitution at position 17, and you can see in this old-fashioned Sanger trace that the blood cells show the normal sequence, but the brain shows a little extra peak. And that represents this mutation being present in about a third of the cells from the hemisphere that we received from the neurosurgeons and were able to study in the laboratory. And as far as we know, it's absent from the rest of his body. So this would be a mosaic mutation limited to the brain, and in fact limited to half of the brain. And this is a known cancer-associated mutation, although AKT3 is an isoform of AKT, which is mostly brain-specific. So this is not a major oncogene. Although the exact paralogous mutation in AKT1 is a major oncogenic mutation. And this causes constitutive activation. So it's a gain-of-function mutation of this kinase. And so in the decade or so since that, it's now conventional that these overgrowth lesions of the brain that are intensely epileptic, whether they're big like Dante's or smaller like this lesion, represent activating mutations. And the vast majority of these cases have activating mutations in this one biochemical pathway called the Mtor pathway. And this is actually the most common cause of intractable childhood epilepsy that is treated surgically. And in fact, these kids generally respond very well to surgery, with more than half of them becoming seizure-free. But this is even though this is, and the mutations that cause this condition are classic oncogenic mutations, activating mutations in PI3 kinase, activating mutations in Mtor, or loss of function in negative regulators. And despite the fact that these are classic oncogenic mutations, these lesions are not tumors. They don't grow. They are stable, as far as we know, throughout life. They do show overgrowth, and so they do show an effective clonal selection, a positive clonal selection of the mutant cells. And then the difference in the size of the lesion seems to reflect merely the timing at which the mutation occurs. The exact same mutations can be found in these smaller lesions called focal cortical dysplasias, or in the larger lesions. And so we presume it's all about timing. In fact, many of these same mutations can cause overgrowth of non-CNS tissues, as described by Les Biesecker and others. They can cause vascular overgrowth, for example. And then the other thing to note is that the higher the variant frequency, and the larger the lesion in general, and that also correlates with the more severe neurological disability, earlier onset of seizures and greater disability. However, the most common sort of lesion are these smaller ones that are only 1 or 2 centimeters in size and that respond very well to surgical removal. And the mutation is typically present in only about 1% of the cells. And so it's remarkable how irritating that very, very small population of mutant cells can be. And again, also the discovery of these mutations has prompted the repurposing of Mtor inhibitors developed for the treatment of cancer for the use of intractable epilepsy, although the results have not been quite as positive as we'd hoped. And I also want to acknowledge Alyssa DeGamma, a former student who worked with Anne on a lot of these studies. And also there's been important work from many labs. As mentioned last, Stephanie Baulach in Paris, Bill Dobbins, Joe Gleason, and Jung Ho Lee in Korea have all done great work on this. So that's really just an introduction. Now I want to turn to adult epilepsy. What happens in adult epilepsy? And the most common form of focal epilepsy in adults is mesial temporal lobe epilepsy. And mesial is just a fancy name for medial, and I'll refer to it as MTLE. And so this is the most common focal adult epilepsy, and this has been described literally for millennia. Hippocrates wrote about, or a treatise that's attributed to Hippocrates, criticized a concept in his day and age that this was the sacred disease. Some of these people with temporal lobe epilepsy were thought to be possessed by spirits because the presentation of temporal lobe epilepsy is typically more psychiatric than it is neurological. The auras can be memories or voices or smells rather than convulsions. But the interesting thing about this is that the age of onset is typically not in childhood, but in adulthood, after a latency period that can be many decades. And then, as I say, after the auras, there can be speech arrest, loss of attention, and then if there are convulsions, they generally reflect generalization of the epilepsy or the seizure beyond the temporal lobe itself. This accounts for 2/3 of intractable adult epilepsy. And it's been known, and it's never been explained why the temporal lobe of all of the parts of the brain are preferentially affected. Those pediatric conditions I just mentioned affect the neocortex, but not particularly the temporal lobe, whereas the adult form preferentially affects the temporal lobe. And we know that it affects the temporal lobe because it has a characteristic neuropathological hallmark. This is called mesial temporal sclerosis. And so if we slice that temporal lobe, the medial structures of the temporal lobe represent the hippocampus, and then the lateral temporal lobe represents the auditory neocortex. And within the hippocampus, patients with mesial temporal lobe epilepsy characteristically show a loss of the neurons in the Ammon's-Horn field or the CA fields. You can see that normally these neurons stain brown for NUN immunoreactivity, and in a typical epileptic specimen, the neurons in these regions are completely lost. And this can be seen radiographically as a little bright spot. And it's known that patients with this particular pathology and this particular radiology respond, again, very well to temporal lobe removal. And so this is the classic form of complex epilepsy. There are no Mendelian forms. There are well-known non-genetic risk factors. 2/3 of patients will have a history of head trauma or of a severe seizure, a single seizure in childhood. And inherited genes so far have been shown to, whether rare or common, have been shown to have little or no role. And yet temporal lobe removal has been known since the days of Wilder Penfield to produce dramatic improvement. And so this prompted us to check whether clonal somatic variants might be important in temporal lobe epilepsy. And this was tackled by Sattar Kushku, an epilepsy fellow at the Brigham, who is just in the process of starting his own lab there. And so initially we collected all the specimens we could find from Boston Children's Hospital, and then we struck up a collaboration with Chris Kale, who was in the process of moving from Yale to Mass General. And he had a large collection of specimens from Dennis Spencer's, from his and Dennis Spencer's operations there. And we performed deep whole exome sequencing, so much deeper, more than 10 times deeper than a typical coverage you would do to look for germline mutations, because the mosaic mutations are harder to detect. They're only in a fraction of the reads, and so you have to get a lot deeper read coverage. And in fact, this 500x exome sequencing is actually has very poor sensitivity, even at that depth, to detect somatic mutations. So in those 100 or so cases, we found a grand total of only 11 somatic mutations that were predicted to have a deleterious effect on the protein, and yet 10 of 11 of these were in the RAS-RAF MAP kinase pathway. And in fact, all of them were predicted to be activating mutations. And this included classic cancer mutations, the KRAS G12V, and another substitution at that same location, the classic BRAF V600E allele, activating mutations in PTPN11, and then loss of function and neurofibromatosis, the negative regulator, and such that these are all predicted to be activating. And so that shows that RAS mutations might be important, or are important in some cases, but we wanted to get a better sense of just how frequent these mutations are. And for that, we needed much more sensitive sequencing. We performed sequencing greater than 1,000x coverage, and Sattar did an amazing job of getting cases from all over the world, from Paris, from Toronto, and sequencing more than 500 cases. And here we find that about 40% of the cases have activating mutations in the RAS pathway. Most commonly in PTPN11, which encodes SHP2, where we see a dozen different patients that all have substitutions at alanine 72, another dozen with mutations at E76, another dozen with substitutions at P498. So more than 100 cases with just PTPN11 mutations, which is 15% of all of the cases. All of them are missense. Many of them are recurrent, and most of them are already known to be activating, either in the context of cancer or in the context of Noonan syndrome, where activation of the RAS pathway is also implicated. Another 100 cases had other activating mutations elsewhere in the RAS pathway, like RAS itself RAS itself, or PTPN11, or MEK itself, or BRAF, although BRAF was relatively uncommon. And this yield actually is comparable to what we would find if we did a similar panel sequencing experiment on these focal cortical dysplasias. And yet, when the most sensitive sequencing methods are applied to these focal cortical dysplasias, you can recover activating mTOR mutations in 80 or 90% of the cases. And so we suspect that these activating RAS mutations might be important in a very large fraction of temporal lobe epilepsy. And of our cases, 3/4 of the mutation-positive cases showed nothing but this mesial temporal sclerosis. They showed no tumor, no dysplasia. In fact, the yield of activating RAS mutations was higher in patients that had no other pathology than it was in those patients that had dual pathology, that had additional things like a tumor or dysplasia. And so in some of these cases, we had matched tissue from the hippocampus and from the neighboring temporal lobe. And what we noticed is that in all cases, the variant is present in a higher fraction of cells, at what we call a higher variant allele frequency, in the hippocampus than it is in the neighboring temporal cortex. So in this case, for example, it's present at a variant allele frequency of 2.5, which means it's in about 5% of the cells. Double the variant allele frequency gives you the fraction of cells carrying that variant. And then it's undetectable in the neighboring temporal neocortex. And then the other thing we noticed is that the higher this variant allele frequency, that correlates with earlier age at the time of surgery. You can see that here, this 50-year-old female had a variant allele frequency of 1, yet a 10-year-old female had a variant allele frequency that's about 8, or a male who was operated on younger than 10 had a variant allele frequency of about 30, and so on. And so it seems like, just like with Mtor, the larger the fraction of cells that carry the mutation, the earlier the onset of the epilepsy, and likely the more severe they are. Well, the involvement of these mutations in the hippocampus creates a remarkable situation, because unlike the neocortex, where I told you all of the neurons are postmitotic, in fact, in the hippocampus, specifically in the dentate gyrus of the hippocampus, this is the one place in the cerebral cortex where stem cells persist throughout fetal gestation and even after birth, and where neurons continue to be formed, at least for a few years after birth. And at the present in humans, it's controversial whether this postnatal neurogenesis in the dentate gyrus is lifelong or whether it only persists for a few years, but there's a consensus that that there's no doubt that it continues at least for the first few years of life. And in animal models, this hippocampal neurogenesis has been implicated in memory formation and consolidation, but it's also modulated by almost everything: activity on the treadmill, toxins, seizures, which induce a wave of neurogenesis in the dentate gyrus, head trauma, which also induces a wave of neurogenesis in the dentate gyrus, and many other things. And so this suggests the possibility that life events might actually modulate the risk of temporal lobe epilepsy by modulating this process of postnatal neurogenesis. And this cartoon explains the way we think about it. So congenital activation of the RAS pathway in neurons is known to be inherently epileptic, as is activation of the Mtor pathway in neurons. Both of these tend to make those neurons that carry the variant intensely epileptic. And so neurons might be born, a small clone of neurons might carry an activating RAS mutation, and this represents a latent risk factor, which might be present in people that never go on to have seizures. But then if there's a risk factor, that would stimulate a wave of proliferation in the stem cells that also carry this variant, and any sort of proliferation is going to favor clones that have activating cancer mutations. And so that would cause the RAS-activated clones to grow bigger than the normal clones. And that would create a higher fraction of cells that are now carrying the mutation. And I just told you that the higher the fraction of cells that carry the variant, the earlier the age of onset of seizures and the greater the risk of epilepsy. And so this model can explain several things. It can explain, in the first place, the predilection of epilepsy for the temporal lobe in the first place because of this phenomenon of postnatal neurogenesis, and also the well-known clinical pearl that seizures beget seizures, that a patient might have his first seizure at age 15, but then his second seizure might be 6 months later, his third seizure might be 6 weeks later, his fourth seizure might be 6 days later, and there's something about seizures that are themselves epileptogenic. And we have some evidence for this model already. We can use the tools that have been developed to look at cancer driver mutations to ask whether these activating RAS mutations actually are under clonal selection in the temporal lobe. And sure enough, in the epileptic brain, they are, as shown by these genes that are above this P of 0.05 significance line. Whereas in the normal brain, they don't show strong evidence of this positive selection. Or we've also taken engineered iPS lines that have one of the activating PTPN11 mutations. We've mixed it 50-50 with wild-type cells, and you can see that they very quickly overgrow the culture. So they clearly have a proliferative advantage. So just to summarize, we think that this has really important therapeutic implications because just as there are Mtor inhibitors, there are also inhibitors of the MAP kinase pathway that have been developed for cancer, but now they actually have potential applicability to, again, a very common form of adult epilepsy. Now, we also want to know why these neurons die. So temporal lobe epilepsy is a degenerative condition. Is this due to the mutation in the neurons, or is it due to the mutation in the glial cells that might kill the neurons? And we think we have some evidence that the mutation is clearly arising in the neural epithelium, because if we sort different subtypes of cells, we can find the mutation in the neurons and in the oligodendrocytes and in the astrocytes. And so clearly it's present in the neuroepithelium. And so we suspect that that is where its origin is. But it's again, present in multiple cell types. And so we don't yet know whether the neurons die by suicide or whether they get murdered. We also would like to formally test this idea that epilepsy induces proliferation in human temporal lobe tissue. And we think we might be able to do that with some single-cell methods that I'll describe later in the talk. So after finding these activating cancer mutations in the temporal lobe and realizing that they could be subject to selection if the cells undergo proliferation, we wondered whether this idea of clonal selection might be important in other diseases that occur late in life and where we know that there is inflammation which also stimulates proliferation and which therefore might also serve as a vehicle to select for mutant clones that might have cancer mutations. And so two brave postdocs, August Huang, a bioinformatician shared with Alice Lee's lab, and then Zinan Zhou, a very talented postdoc, decided that we would take a postmortem Alzheimer brain and to see if they also had activating cancer mutations in the cells of Alzheimer brain. We took prefrontal cortex from almost 200 Alzheimer cases and over 100 aged controls. And we did a cancer panel of 149 cancer genes where we could detect mutations even if they're present only in 1 or 2% of the cells. So we sequenced it even deeper, almost 2,000x average coverage, similar in Alzheimer's and controls. And we found, sure enough, the Alzheimer cases actually have a higher rate of mutations in cancer genes than age-matched controls. The age-matched controls have plenty of them, but Alzheimer has, if we use our stringent pipeline, which basically eliminates recurrently mutated genes, you see that the difference is about 30% increased, and it's a more modest increase if we include all mutated genes on the panel, but actually a higher p-value. And this difference is robust to controlling for gender and age, sequencing coverage, postmortem interval. And it appears that this accumulation of cancer driver mutations is something that happens with age, but that is enhanced in the Alzheimer brain. And perhaps modestly, although it's particularly strongly enhanced when you look at the genes that are recurrently mutated. Those show a much stronger enrichment compared to control. And the ApoE4 allele seems like it modestly increases the rate of somatic mutations, both in the Alzheimer brain and in the normal brain, although these differences are not statistically significant. But this might be a way of thinking about how ApoE4 creates risk by stimulating this process of clonal selection. So this is a list of the most commonly mutated genes, and it's not RAS this time, it's not Mtor. In fact, it's genes like DNMT3A, TET2, and ASXL1. These actually are genes that are very familiar because they are currently mutated in blood disorders, clonal disorders of blood, things like clonal hematopoiesis, myeloid leukemias, and that is consistent with the idea that some cells, particularly the microglia, share some common phenotype and lineages with these myeloid cells of the blood. So you see that there are you see that, again, these genes are mutated both in Alzheimer's and aged controls, but more commonly in Alzheimer's. There are several genes that are nominally have greater numbers of mutations than in Alzheimer's than control, although this doesn't survive correction for 149 different hypotheses. And in Alzheimer's, what we see are individuals that have simultaneous mutation of multiple genes at the same time, as shown by these arrowheads, and we have not yet seen that in a single aged control. And so that is another difference that's statistically significant. And then what we find in Alzheimer's also is that the mutations are present in a larger mutant fraction, suggesting that they're under positive evolutionary selection in Alzheimer's to a greater extent than in controls. This is particularly true for the tumor suppressor genes, and there's a 40% increase for these, again, recurrently mutated genes that I just showed you on that panel. And again, you can demonstrate that these mutations are under positive selection in both normal aged cases, but to a greater extent in Alzheimer's than in normal aging. So we think of this as a phenomenon that happens, this clonal selection happens in normal aging, but is enhanced something about the Alzheimer brain causes it to be enhanced. And so what cell type carries these mutations? Based on the identity of the genes and what we knew about the pathogenesis of Alzheimer's disease, our top suspect was the microglial cells, which are formed from a myeloid-like precursor that separates from the other myeloid lineages very early and then continues to develop in the brain. And so Xenon developed a way to purify these, or at least to enrich them, so that we can get a preparation that's about 3/4 or more microglial cells. And this microglial fraction, for all of the proliferation stimulating mutations that we tested, you can see that it's strongly enriched in microglia compared to other cells. We can't say that it's completely absent in the other cell types, but because our sorting is not perfect, but clearly they show anywhere from 100-fold enrichment in some cases. And you can see also that some of these mutations are present in a very large fraction of the microglia. So this mutant allele fraction or variant allele fraction here of 22% means that basically that mutation is present in half of the microglia of that person. This one's present in 25% of them, and so on. And so they really can, these clones seem to take over a large fraction of the microglia in some cases. We tested whether these are shared in the blood, and the ones we tested were all shared in the blood. Although we then worked with Sid Jaswal from Stanford, who analyzed a whole exome sequencing of these same cases. And what we find basically is that the variants that have higher allele frequency are shared in blood and brain, but then the variants that presumably occurred later tend to be present in the brain but are not shared in the blood. And so this might be that there's some early progenitor that's shared between brain and blood, but then it looks like the cells, once they're in the brain, go on to develop additional mutations that might contribute to the clonal selection. And so what's the impact of these mutations? So again, by the analogy to clonal hematopoiesis, what happens is these same exact mutations develop in the hematopoietic stem cells, and they create mutant progeny. And as those mutant progeny develop larger and larger numbers of mutations, they develop an increasingly abnormal pattern of transcription. They develop an inflammatory signal, which is thought then to be toxic to other cells. And this is how clonal hematopoiesis is thought to be a risk factor, not just for cancer, but also for heart disease, because it creates an inflammatory environment that contributes to perhaps many different diseases. And so we wanted to know then whether these driver mutations in brain microglia also might induce an inflammatory state. And we approached that in two different ways. First, thanks to Ben Ebert, I was introduced to Irene Papapedrou at Mount Sinai and her colleague Samuela Morrow, and they had already targeted our top 3 genes, TET2, DNMT3A, and ASXL1 in iPS cells, and then differentiated them into microglial cells. So we set about to collaborate. And they basically then differentiated these to a microglial state and measured their transcriptome. And what we find is that all 3 of these genes contribute to abnormal inflammatory patterns that resemble a disease-associated microglial signature that's been described in Alzheimer's brain. It's a subset of microglia that take on a very inflammatory pattern of transcription. And we see that reproduced notably by DNMT3A and ASXL1 mutations. And then we also see a proliferative signal, which is stimulated by ASXL1 and TET2 mutations. And Maya Talukdar is a really talented MD PhD student in my lab who basically has analyzed these data. And this specifically shows that these engineered microglial cells have upregulation of a host of inflammatory genes and inflammatory signatures and also proliferation signatures. So so these mutations create a very abnormal transcriptome in the microglia, which is very consistent with the abnormal transcriptome that's seen in the microglia in late-stage Alzheimer's disease. A second approach we took was actually focusing on the brains themselves. We can't call the point mutations in single-cell RNA-seq data, but we can call copy number variants. And so Maya basically, there are also copy number variants that can drive clonal hematopoiesis. There are particular recurrent parts of the genome that actually are recurrently mutated to push CHIP just the same way point mutations can. And so we could call these chip-associated copy number variants, or mosaic chromosome alterations. And we did this in a very small sample size of just 30 Alzheimer cases and 30 aged controls. But we found that there are a handful of mosaic chromosome alterations in Alzheimer's disease and a nominally larger number than in aged controls, although this is not statistically significant because of the small sample size. And again, you see a very large number of mutant microglia in Alzheimer's compared to control. And the cells that preferentially carry these mosaic chromosome alterations are, again, microglia or capillary associated macrophages, and less commonly oligodendrocytes, but then not OPCs or neurons. And again, those cells that carry these mosaic chromosome alterations again have this abnormal pattern of transcription that resembles these disease-associated microglia. We see upregulation of many individual genes that are a component of the DAM signature, and then we see upregulation again of inflammatory pathways like response to cytokines and so on, or cellular response to interleukin-4. And so our way of thinking about this is that I think it's accepted that the Alzheimer brain is an inflammatory environment and that there is proliferation of microglial cells at a higher rate than normal. And whenever we see inflammation and proliferation, that is going to drive evolutionary selection in favor of cells that carry oncogenes, because those cells will survive better and will proliferate better than wild-type cells. And so that proliferation then will invariably select for driver mutant clones. And so this creates a phenomenon we call microglial clonal expansion, or MICE, because of the survival advantages of these cells. But again, the survival advantages of those cells comes with a cost, because those mutant microglia have inflammatory signatures that have been shown in blood cells to promote the death of their neighboring cells by secretion of interleukins and other cytokines that are toxic to other cells. So in the last 10 minutes or so, I'll tell you a little bit about what happens to a neuron's genome in some of these conditions, with aging and with disease. And basically, this develops from isolating single neurons in the brain and then sequencing their genomes and comparing them. We had to do this because the clonal structure of the brain is so complicated that the only way of studying the clonal structure is to focus on single neurons. And then we found that this got us into a whole area of looking at the aging genome of single neurons. And so basically we take single nuclei from typically the prefrontal cortex. We separate neuronal nuclei and put them into single wells. You can amplify the genome by a variety of different methods and then sequence it the way you might sequence your genome from your blood DNA. And Mike Lodato and Rachel Roden led a study that was published in 2018 where they looked at single neurons from a range of ages, anywhere from infant to aged individuals, and we would just take like 3 or 4 neurons, literally, from the prefrontal cortex of an individual. And when you do that to a newborn, you find that there are hundreds of single nucleotide variants. In other words, point mutations, a substitution of one nucleotide for another in that single neuron compared to the germline of that same person. But then remarkably, in teenagers, it's closer to 1,000, and in middle-aged individuals, it's 1,500, and in elderly individuals, it's more than 2,000. So since then, there are better ways and more accurate ways of amplifying the genome of single neurons. That MDA method we originally used creates exponential amplification, but results in a very unequal amplification of the genome and a fair amount of amplification artifact. Chuck Gawad at Stanford developed a more linear method called primary template amplification, where the pieces are shorter and much more even, and it gives remarkably even amplification of the genome. And with this method, we find that this age-related accumulation of mutations is a little slower. Instead of 23 mutations per year, it's 17 mutations per year, but it's incredibly linear. And so you see 17 mutations in the very first year of life and 17 mutations in the last year of life. And that there's very little variability between different cells from the same individual in a normal person. And this is work that was done by Joe Luquette recently. Mike Miller and Xenon Joe from my lab, and then Joe, who's in Peter Park's lab, was published a couple of years ago. And that work has now been replicated in 4 different labs using 5 different methods. All find just about 17 or 18 SNVs per year. This is work from Sunny Shi that used transposases. There's a study from Inigo Martin-Carena at the Sanger Center using duplex sequencing, and an unpublished one from Gilad Avroni's lab. They all show I've taken the liberty of taking some of the points from the other papers and putting them all on the same plot just to show how incredibly precise and linear this is with multiple different methods. And so how in the world do non-cycling neurons develop double-stranded point mutations? And I don't know the answer to that, but I'll just propose a plausible mechanism because I find it a little easier to think about it. And that starts with the fact that DNA damage is ubiquitous. It's generally damage to one strand or the other, or else there can be double-stranded breaks. At any given moment, there are more than 1,000 single-stranded lesions in the genome. You can see the different types here and how frequent they are. And these are repaired by a variety of different mechanisms, but one of the mechanisms is nucleotide excision repair. But the way that works is by actually excising 29 base pairs and then repairing it by replicating the undamaged strand. But if a cell happens to have 2 lesions within 29 base pairs of each other, then it's actually repairing one strand by using a damaged template, and so that would then create a double-stranded change. So this is at least one mechanism. So even the non-replicating cells are actually replicating their genome just 29 base pairs at a time. And in fact, given the frequency of these single-stranded events and the low rate of 17 or 18 per year, there are more than enough, you could predict that there are more than enough times when you would have 2 different lesions within 29 base pairs of one another to account for what we see. So what happens then in disease? And so Mike Miller and Mike Lodato, we collaborated then with Alice Lee's lab to study single neurons from postmortem Alzheimer brain. We focused on large new-end-positive neuronal nuclei, and these are preferentially pyramidal neurons, which are preferentially affected by the disease. And we find that these neurons have about 200 extra mutations compared to age-matched controls. And that's more or less equivalent to the 10 years of life that you lose to Alzheimer's disease if you figure that you accumulate about 17 or 18 mutations per year for 10 years. And so what causes them? So to do that, we can again use tools developed for the study of cancer that look at base substitutions and then look at the 3 base pair context, the 2 neighboring bases in which that particular substitution occurs. So here's a C to A change, and then there might be an A on either side of it. And so this creates 16 different possible C to A changes. And then it looks for recurrent patterns of substitution. And it turns out that things that damage the genome never do it in a random way. They always preferentially affect some bases and not others. So this is a simulated lung cancer genome where you sequence it and get all of the somatic mutations, and then you can sort them into recurrent buckets using mathematical methods like non-negative matrix factorization. And so in this simulated lung cancer, there are several recurrent patterns. One has red C to T changes, particularly at CpG dinucleotides, and this is called single base substitution 1 or signature 1, and this is seen in all dividing cells because this represents cytosine deamination during the cell cycle. Or there's one called signature 4, which has these blue C to A changes, and that's only seen in lung cancers of smokers or head and neck cancers of smokers because this is due to direct toxicity of cigarette smoke on the genome. And then there's another signature called SBS5, which has a more broad pattern of nucleotide substitution, but particularly C to T and T to C. And this is actually represents just aging. It's seen in all tumors and in all tissues, and is proportional to nothing but the age of the patient at the time of diagnosis. So in neurons, we set our own signatures. We used letters to refer to them rather than numbers. And then after the fact, saw how they compared to the cancer signatures. And the age-related accumulation in neurons is mainly driven by a signature we call signature A, but this resembles very closely the cancer signature SBS5, which is this mysterious age-related signature, which is, again, as I said, proportional to nothing else other than the age of the patient at the time of diagnosis. But here we show that this is actually completely ubiquitous. It even happens in cells that don't undergo cell division. And we actually did a paper on cardiomyocytes and showed that non-cycling cardiomyocytes also accumulate this SBS5 signature. And so again, it may be consistent with some sort of faulty nucleotide excision repair. And so then what happens in Alzheimer's disease? What about these extra mutations? Well, we find that these extra mutations actually represent the enrichment of a mutation pattern which is rare in the normal brain, which we called signature C, and it has these C to A changes and these T to A changes, which are rare in normal brain. And we find that, in fact, this signature behaves very differently than the normal neuronal signature. The normal neuronal signature, signature A, is very proportional to gene expression. It happens more commonly in highly expressed genes and is uncommon in genes that have low expression. But this extra signature in Alzheimer's does not show that same relationship at all. It's not proportional to gene expression. And so these signatures can sometimes implicate mechanisms of toxicity. We suspect that these C to A changes might represent oxoguanine because that's thought to see what happens in cigarette smoke. And in fact, you can see that there's an excess of oxoguanine. You can stain it immunohistochemically in Alzheimer neurons. It increases normally with age, but then is further increased in Alzheimer's disease. More recently, Xenon's looked at frontotemporal dementia. And specifically due to cases where there's an amplification of the C9orf repeat. And that shows a very similar phenomenon to Alzheimer's, about 200 extra mutations, and actually with a similar pattern of nucleotide substitution. Now, with the better techniques, we can also analyze a second type of mutation in neurons called indels. So indels are the insertion of a couple of bases. These are typically short, just a few base pairs, or the deletion of a couple of bases. So I just refer to them as indels or somatic indels, S-indels. And these accumulate fairly linearly in neurons as well, but almost 10 times slower than SNVs, only 2 or 3 per year. They have actually an even stronger relationship to transcription than the SNVs. So that both indels and SNVs are enriched in highly expressed genes. That's, again, shown here for SNVs and here for indels. But the indels show a stronger enrichment. And again, they're both also strongly enriched at neural promoters and neural enhancers, but particularly the indels. Again, the SNVs show some enrichment at H3K27 acetylation peaks. The indels show an even stronger enrichment. The SNVs show a little enrichment at transcription start sites, but the indels are very, very highly enriched at transcription start sites. So the indels particularly happen exactly where you don't want them. They happen in enhancers. They're enriched in enhancers. They're enriched in promoters. They're enriched in any form of open chromatin. And so they are very damaging. And that these 2 or 3 indels per year are actually more damaging than the 15 or 20 SNVs once you actually figure where they occur in the gene, whether they occur in a gene or outside of a gene, and whether they actually are likely to impair the function of that gene. So recently, work that's not published yet we found this several years ago, but didn't know what to do with it. Both Alzheimer and ALS FTD actually have a very large increase in these indels. And it's very spotty. So this shows age-matched controls, and then Alzheimer's single neurons and ALS FTD single neurons. And you see that some neurons look approximately normal, and this is a log plot. So this is 5,000 here, and this is 500. But you see some neurons have thousands of these indels, which would be incredibly damaging for a large number of genes. And they happen in a very distinctive pattern. So if you look at all of the indels in normal neurons, you see a fairly wide distribution of different types. These orange ones are 1 base pair deletions. These green ones are 1 base pair insertions. And then you have 2 base pair deletions. You have 2 base pair insertions in blue, and so on. In these abnormal ones are almost exclusively 2 base pair deletions. This shows, again, I've reduced the normal, because you see this is the normal pattern, and then superimposed on this is this huge spikes in ALS and in Alzheimer's disease of these 2 base pair deletions that occur in a highly recurrent pattern in both, and a very similar pattern, of essentially an indistinguishable pattern in these 2 very, very different diseases. Now, we didn't know whether these were single-stranded events that were just a lesion on one strand and not the other, or a double-stranded event. We suspected they might be single-stranded because Inigo Martin Carrena had also studied Alzheimer neurons, and he'd seen many similar patterns of indels, but had not seen this very large accumulation of these indels in Alzheimer's disease. And so we thought, well, they must be single-stranded, but in fact, Diane Shao adapted this method of tagging the two different strands of DNA. This is called Meta-CS, developed in Sunny Shi's lab, where you use transposases to label the Watson and the Crick strands separately. And so if a variant is present, is truly double-stranded, it comes through both on the Watson strand and the Crick strand, and so you can do a complementarity check. You can be certain that this is a true double-stranded mutation. Whereas if there's any sort of amplification artifact or any sort of post-mortem change, this will be present on one strand and not the other, and so you can filter these out. So what we find is that these two base pair insertions are most commonly single-stranded, but in fact they are definitely frequently double-stranded as well. And so this makes us, so clearly they are real, but in fact this makes us believe that these single-stranded events are very likely to be a biological phenomenon because of their specificity and their resemblance to the double-stranded signature. So I'll just wrap up here. The neuronal genome is inherently changing. The neurons gain an SNV every couple of weeks throughout your life and 2 to 3 indels per year in a remarkably linear fashion normally. The mutations are enriched in exactly the most vulnerable parts of the genome and transcribed parts of the genome. And then AD and ALS both show increased numbers of single nucleotide variants compared to normal neurons, about 200, equivalent to the 10 years or so of aging that is lost to either of these conditions. But then, perhaps more remarkably, they show these large increases of hundreds to even sometimes thousands of indels. And these, again, are highly enriched in genes. And the mechanism we don't yet understand, but we're very, very actively trying to figure out what the mechanism of there are, because this could potentially cause widespread changes in gene expression that might ultimately have some relationship to the death of those neurons. So, I'll just conclude by saying that this work on somatic mosaicism has really changed the way I think about every age-related disease of every organ. The diversity in any one cell type of your body is greater than the genetic diversity of all of the 8 billion people in the world in a germline level. So I'll just illustrate that with a simulation. So we have 80 billion neurons and 80 billion glial cells approximately. And each of these are born, as I told you, with about 100 somatic SNVs that are these developmental clonal ones from the cell that occurs during the cell divisions that generate it. And so that gives you 8 trillion somatic mutations just in your brain neurons. And of course, now the genome starts looking pretty small. It's a mere 3 billion base pairs. And so 8 trillion divided by 3 billion is 2,000 times. The average spot in your genome is mutated 2,000 times just in your neurons, just at birth, to say nothing of the age-related ones I was just telling you about. And then a similar calculation will hold for whatever your favorite cell type is. The same calculation holds for glial cells. So glial cells already have a mutation in every single, not just every gene in the genome, but every nucleotide in the genome. And it's just a question then of whether they're under the right sort of clonal selection to cause those mutations to have an effect. And this simulation is copied from Ben Ebert, who described this for 10 billion T cells. And so then I believe that any inflammation is going to breed proliferation. And any kind of proliferation is going to breed a competition, a clonal selection of those cells that happen to have activating mutations that are essentially cancer-like mutations. But oftentimes, these cancer mutations occur in cells, like neurons and microglia, that don't form tumors, but they create alternate disease states that don't look like masses that you can take out. And so, again, other mysterious non-neurologic diseases are likely to reflect other kinds of roles of somatic mutation. So I tried to acknowledge everyone as I went. The single neuron aging work was started by Mike Lodato, and then Mike Miller and Xenon played a major role. The Alzheimer's disease work, again, Mike Miller and Mike Lodato and August Huang. And we've been really lucky to collaborate with Brad Hyman from the MGH Alzheimer's Center. The temporal lobe epilepsy work, really the work of Sattar Kushku. Oh, I didn't mention Yilan Wang, who is the bioinformatician from Alice Lee's lab with which we did that work. And then the clonal mutations, I think I mentioned all these people here, but we've been really, again, lucky to have the cooperation of David Bennett from the ROSMAP project, and then Xenon, the FTD ALS. And would be no place without Peter Park, a brilliant bioinformatic collaborator. I want to particularly shout out Joe Luquette, who is involved in many of these projects and is just like a genius when it comes to analyzing these things. And then Alice Lee's lab, on which we collaborate with some of the other studies. Again, really, really bright people in her lab. So thanks very much, and I'll be happy to answer questions.
Repeat this. There we go. Okay, bang. This came in early as we're waiting. Earlier question from any information about the existence of these mutations in the population of children who have experienced a febrile seizure?
We don't really have any that specific information since we can only really work with either a postmortem brain or a temporal lobe resection. What we're trying to do, though, is to figure out if those, if our patients that have a history of febrile seizures, if what I'm saying is true, we hypothesize that seizure would have induced a wave of neurogenesis in the temporal lobe. Mm-hmm. And, I mentioned that these cancer signatures are very specific. And so one of them is specific for cell division. When I go back to this lung cancer, this SBS1, is seen only in dividing cells, and it's not present in neurons. The neurons have a few of these mutations from their development, but then they don't accumulate them with age. They accumulate other things. However, we can now look in the temporal lobe specimens to see if they have more of this signature than normal neurons, and that would be an indicator that those neurons had undergone additional rounds of proliferation. And so we'd like to be able to correlate that with their history of risk factors.
Okay. I'll move on to one more. In cases of clonal proliferations in these neurological diseases due to somatic variants, should these cases also be described as benign neoplasms?
Let's see. I guess it depends on the precise definition of the term neoplasm. I'm trying to recall exactly what I was taught in pathology, but that was 40 years ago or 35 years ago. My understanding of a neoplasm is that they grow. And so by that definition, no. Because, again, the pediatric ones are fixed disorders that, as far as we know, don't grow. Probably we'll find out someday that they are growing in some ways where their overall mass or appearance doesn't change. They're probably a lot more dynamic than I'm giving them credit for, but they don't obviously grow in the way I typically think of as a neoplasm.
Hi, very nice talk. So I think your epilepsy data suggests that because of the networked nature of neurons, a very small proportion of affected neurons can have an outsized effect.
Right.
And so I'm just wondering if you have thoughts as to how or whether these somatic mutations may contribute to normal human diversity in terms of neurocognitive function and behavior.
Yeah. Thanks very much, Chris, for that question. So I think it's very likely that they do, although it's a little hard to know how to, make that argument. And the reason I think it's likely that they do is that developmental somatic mutations that are clonally shared are common enough so that a typical person, if we go through the math, a typical person will contain a function-altering exonic somatic mutation in like about 2% of their cells or more. So basically all of us have a function-altering exonic mutation in some clone in our brain somewhere, to say nothing of the ones that might be also function-altering but non-coding. And so, there are so many of them that they are very common. And as I say, once you get down to small clones, basically we all have everything. So I do believe that, but again, how to correlate them with differences in behavior or unique aspects of people. And what's really cool is that the developmental ones actually are enriched in non-coding regulatory elements, enhancers and promoters. And what's interesting about that is that exonic mutations are pretty much all bad. The protein sequence of all proteins is optimized for perfect function. A mutation is not likely to improve the function of a protein. On the other hand, mutations in enhancers leave the protein coding part of the gene intact. And so they might shift its expression. They might upregulate it or downregulate it. They might turn it on someplace where it's not normally turned on. So those are just the kind of mutations that might generate functional diversity without creating disease. And so I'm really interested, whether Einstein's brain, had some interesting enhancer mutation and so had some gene turned on, a place where it might not normally be. Sure.
Oh, yeah, please.
Go ahead.
Sure. So 2 questions. The first one is, yesterday I went for a 2-mile walk with a friend of mine. She's almost 98 years old, is almost never sick. She exercises at 8 in the morning, all these good things. And when I look at these horrendous numbers, I ask myself, how can she possibly avoid all these debilitating mutations, really?
Yeah. Right.
Yeah, that's it,
Yeah.
She's amazing. How does that happen?
Right. Well, obviously that's a great question. I would like to learn the answer right away before I get one day older. What fascinates me about these somatic mutations in microglia and things like that is it's a potential way of integrating our inherited genome with our lifestyle. Because these many things might. So we're doing a parallel study of chronic traumatic encephalopathy in American football players. In fact, with Ann McKee in Boston, who has analyzed the brains of many famous football players, and so we see similar patterns of mutation in the genomes of single neurons, and we have some evidence that they also have these clonal somatic evolution as well. And so you can imagine that things like lifestyle changes or head trauma might induce inflammation in the brain, and again, that would then stimulate proliferation of these cells. And so that's a mechanism how these lifestyle events might integrate themselves by potentially promoting in the case of disease, proliferation and clonal selection of cells that have damaging mutations.
Which sort of relates to the second lesser question. Early on, you mentioned something very, very briefly about treadmill activity, and I was wondering, how does that relate to a mutation in the brain?
So as far as we know, it doesn't create mutations in the brain, but it creates a roundup proliferation in the dentate gyrus of the hippocampus. And in animal models, they've studied that treadmill running, and they've shown that if they take a run on the treadmill, they get a wave of neurogenesis in the hippocampus, and that seems to help them learn. And so, that's generally thought of as a positive thing. I don't know exactly whether, as far as we know, treadmill running is not a risk factor for temporal lobe epilepsy. So I haven't totally figured out how the positive aspects of hippocampal neurogenesis, might integrate with the potential for it to induce clonal selection of damaging variants.
Okay, one more question to close if you're still holding up. I want to repeat the CME code that folks at home can't quite see, 50117. So could the enrichment of mutations in highly transcribed genes be a result of transcription complex creating the mutations?
Yes, it's possible that if I understand correctly, the person is suggesting that the transcription complex itself might induce a mutation. Yeah. The way we generally think about it is that it's something about the process of transcription. It need not be anything more than just opening the chromatin and exposing the base pairs of DNA, which are normally protected by the phosphate backbone, as they open up, they have to open up to be transcribed, and so then that leaves the bases vulnerable to the sorts of reactive oxygen and things that are ubiquitous.
Okay, very good. Any closing?
No. Just a big thank you, I think.
Great,
thank you very
thanks.
much.